A while ago, I talked about the anatomy of the human body and different body parts, but I didn't talk about how dissection is done. Dissection. Dissection is a method used in the field of anatomy to observe and operate surgery on a specific organ or a body part. Surgery. To perform surgery, we use certain specialized surgical equipment to operate on a body or a body part. So in today's video, I will perform dissection on one animal specimen like a lizard or a frog and on one plant specimen like a flower and a leaf. To perform dissection, I need a dissection kit, except I don't possess any proper information or equipment on how to do it. So I went and got out this one, which was the only one I had. So I went and got out my biological specimens and I unlocked all of them. What I had was a black beetle, a spider, a snail, a cockroach, and a cricket, and finally a lizard. As you can see, it's already been dissected. But anyway, I took it out of the bottle containing propanol, which is used to store specimens for longer periods of time, and I placed it on my dissection tray. Over here we can see its dragon eyes, along with its long tail, body containing fangs, which let it stick to targets and surfaces. So I took a scalpel and made an incision right in its abdomen to reveal its internal organs. I removed the inner layers of the skin as much as I could. Here we can see its ribs. I tried to open it up with various incisions as I used some forceps to remove as much inner tissue as possible. But turns out it actually came out hollow. I don't know what had gone wrong, as I was hoping to see at least the heart, kidneys, and intestines, but instead it came out hollow. Anyways, if to see the organs, I might want to dissect a frog, but I don't have one right now. In future, I might probably dissect a frog, and if you want to see it, then definitely let me know down in the comments. Well, this was all about the dissection of an animal specimen. Now I want to dissect a plant specimen. To start with, over here I have a specimen of leaf and a bunch of flowers. The plant reproduce and bear fruit through a process called pollination. The flowers are the reproductive part of the plant, comprising of four main parts: sepals, petals, stamens, and carpels. The pistil is the female reproductive part, comprising of stigma, style, and ovaries, while the stamens are the male reproductive part, comprising of anther and filament. The stamen produce pollen grains and they follow up into the stigma, end up in the style, and then into the ovaries, which fertilizes the embryo and forms seed and bear fruit. However, since this is a China rose flower, meaning it doesn't obtain any ovaries, meaning it is just a self-pollinating flower. The leaf consists of three main parts, comprising of lamina, petiole, and the leaf base. The lamina or blade is the flat green surface used to capture light for photosynthesis. The petiole is the narrow stalk connecting the leaf blade to the plant stem. The lower portion of the leaf that attaches directly to the stem is the leaf base. So using a scalpel, I cut the midrib to the apex, exposing the veins of the leaf, and cut two small portions of the sections from the leaf. I then place one piece into some ethanol and the other one on a glass slide. I then place one drop of water onto the specimen and apply a slip, while I took out the alcohol ones and cover that up too. The alcohol ones sucked out all of the moisture, causing the plant cells to become plasmolized, meaning the cell boundaries and organelles will become more visible, while the water ones will make the cells flaccid or turgid in water depending upon the concentration and condition. So I loaded both samples into the microscope and looked at the alcohol ones. Here I can see the cell wall and membrane along with small pigmentations of chloroplast helping in photosynthesis, and at some level of magnification, stomata on the surface of the cuticle, which helps in osmosis in plants. These are some pictures along with the cool demo of microscopy. I have never seen anything like this before and it was so much fun to watch. However, the water ones remained normal and the cells are either filled with water, causing most of the cells to become and appear not properly at this rate of magnification in the area of the microscopy. Anyways, that was all about a leaf specimen, and now I want to dissect a flower specimen. So again, I took out my scalpel and cut the pistil. I also removed the petals and then cut right into the sepals. From the pistil, this was the closer view of the stamen. where the upper part is the filament and the lower part is the anther. This produces pollen grains which then travel to the stigma, fertilizing the ovaries. Now let us see some flower petals under the microscope. So again, using some micro scissors, I cut out small sections which I then place into alcohol and place one drop into another one. In this case, the petals slowly lose color, which means the alcohol destroys the pigmentation containing chromoplast. Upon microscopy, I can see the cells where I can see the cell wall and certain inner organelles or secretions made by the cell. However, upon changing the objective to the other specimen, I can see small little dots of chromoplast, which are the pigmentations giving color to the petals. Here's a small demo. I also had never seen anything like this before. Well, anyways, this was all about the dissection and observation of plant and animal specimens. In near future, I will show dissection on frog, but for now, this is it. If you like watching videos like these, consider subscribing to my channel and liking my videos, and I will hopefully see you in the next one. Thanks for watching.